In our group, we love flowers, but we don't just love them because they are beautiful to look at. Yes, they are beautiful to look at. But if you are interested in diversity and how evolution can create a myriad of form and shape, and how development is going to sculpt that, whether it's an animal or a plant, I would argue that flowering plants are really the star of the show. So I just put a few examples here. You've all seen flower before, but just you see a handful, and you can already appreciate the diversity of shape and form we've got. And this is because flowering plants are a really, really successful group from an evolutionary viewpoint. Nobody knows how many flowering plants there are, maybe something between 300, 400,000. We keep discovering new species. And numbers don't mean much, really. The example I always like to give is if you just look at one family of flowering plant, the orchid, so I've put a few examples here, there is more species of orchid than all the species of mammal and birds together. I love animals, they're fantastic, but again, I think plants are doing a really good job here. Another question that we still haven't answered properly, we don't know why they are so successful. There is probably a combination of factors, it's not just one thing, but one of these factors is most certainly their interaction with pollinating insects, okay? And if you look in evolution, when you have bursts of diversification of insects, very often you often have bursts of diversification in flowering plants. So there is an important love story there. And one story I always tell that I think illustrates this coevolution between plants and the animal that pollinate them is the Christmas orchid. So, or people call it sometimes Darwin orchid. So this is the plant I'm talking about. It's a fairly not particularly patterned flower, it's white. What's interesting is that you have this long extension of one of these modified petals called petal. That's this really long green sock you see. It's called a nectar spur. And you don't appreciate the size here, but the flower, when you take the spur, is like 30, 40 centimeters. It's very, very impressive. And a British grower sent this orchid, I think in 1862, to Darwin, collected from Madagascar. And when Darwin unpacked it, he said there must be an insect somewhere with a tongue long enough to reach. And as often, everybody thought, is completely crazy. There is no such thing as a 30-centimeter tongue insect. And it actually took 40 years for people to identify the Wallace moth, which is actually the pollinating species of this orchid, that has a very, very long tongue. And it's only in the 90s, so over a century later, that people managed to capture this kind of footage and show that actually this insect is a pollinator. So when you like flower and flowering plant, it's not just nice naturalist story like this one. It's also really important because a third of what you eat comes directly from animal-mediated pollination. What this means is every 3 mouthfuls you take, you can only have it because somewhere at some point, most likely an insect, landed on a plant and pollinated it. Okay? And it's not just the food you eat, but also most of the fiber we use for clothing. If you are a carnivore, the animal you eat also rely completely on plant-mediated pollination. And that's why all the examples you choose yesterday actually are all pollinated by animal, and they all rely on animal-mediated pollination. Even, maybe, this guy. So there was a paper a few years ago that argued that brown algae like seaweed and kelp might even be pollinated by this kind of little crustacean. So even this one managed to make it. And this is probably completely fake news. This is a quote that I attributed to Einstein. We're almost certain Einstein never said that, but I like it because I think it captures something really important. It captures our dependency to this tiny animal and this small event of pollination that look like cute thing happening in the garden that you see on Gardener World on TV. But our human society completely depend on this interaction. So how does it work? Well, as all good, successful relationship, it's now important to have a really good communication. And plant and their pollinator have really established all strands of communication to make sure they attract each other efficiently. So animals can rely on olfactory cues, so scent, tactile cue after landing, but of course visual signals are really important. And these visual signals, they can take all sorts of form, but one thing that we know really matter are patterns. So what I mean by flower pattern, petal pattern more specifically, are these colorful motifs you see on the surface of most flowers. And they can take all sorts of shape. You can have some stripe. You can have what we call a bullseye. You hear a lot about bullseye today. This is where the center of the flower contrasts with the periphery. You can have tiny spots. You can have venation. And of course, the range of possibilities is endless because you can combine this pattern to create things that are more and more complicated and elaborated. So why do these patterns matter for plant-pollinator interaction? Well, there is probably two main things to highlight here. The first one is that they can work as what we call pre-landing signals, so signal from the distance to attract the pollinator to the plant in the first place. Either because animal pollinators have innate preferences wired in their DNA that make them particularly attracted to a specific motif. Most likely, it's not that. Most likely is what we call acquired preference. So imagine if you have a bee, you land on a flower, you discover this flower is super sweet because the nectar is particularly rich in sugar, you may develop a really strong affinity for this specific motif. So pollinators are really good at associating a signal with a reward. And this explains why very often, if you have done that yesterday, and in the garden there's lots of them at the moment, all sorts of pollinators. If you track one pollinator, very often you see it visiting one flower type and ignoring everything else. Okay? And this is because of this association created with one specific motif. But the story doesn't stop there. So after landing, this pattern, they act as what we call nectar guide. So what we mean by nectar guide, it could be two things. First, it might be a way of advertising hidden reward. For instance, in this snapdragon flower, the reward is mostly the pollen grain. Pollen provide are a source of protein for pollinator, but the flower is closed, so you don't see the pollen grain inside. So the idea, this splash of yellow you see at the front, is to say, trust me, there are pollen here, just come in. Okay, a bit like an outside advert you would put. And it's also like a roadmap to help the pollinator position itself correctly so it can get to the reward, pollen or nectar, very effectively, but also maximize the efficiency of pollination. And this is one of my favorite examples. So this is research that comes from the US, people working on a plant called Mimulus. So in the flower of Mimulus, you can see there are the two yellow highlights, exactly like what I told you on Snapdragon. And these two highlights are on the ventral petal, and they are really used by the pollinator to orientate itself. So this is the correct way to visit the flower. You come head with the belly of the pollinator facing the yellow highlight. So the back will be in contact with the stamen and the pollen grain. How do we know they use that to orientate themselves? Well, really simply, if you twist the flower, the pollinator enters the flower the other way around. So they really use that, and not gravity, to orientate themselves. Now, this is fun and cute, but the problem is, like, if you use a mutant that lost the yellow highlight, half of the time the pollinator gets it wrong, okay? They choose the direction of entrance completely randomly. And this is a big problem because the yield of seed production, so pollination success, dropped dramatically. So such a tiny difference can have a direct impact on the fitness of the plant. And also people discover interesting things. So some pollinators sometimes are lazy, and instead of landing properly and doing their job, they land at the back, they drill a hole, and they can access nectar. And people show that if you have the spot on this plant, on Alstroemeria, they rarely do it. But if you remove the spot, very often that's what they do. So it's really important to remind them what they are supposed to be doing. So you tell me, okay, plants have flowering plants have pattern on their petal. I've seen plenty that don't. And it's because they're lying to you. They're lying to you because they have pattern, but in the UV part of the spectrum that sadly we can't see, but most pollinators can. Okay? So this is an example. This is silverweed, completely yellow to us. A bee would be able to perceive the contrast between the center that absorbs the UV and the periphery that reflects it. And as I told you, this is because our visual system and our photoreceptor are different from the one of pollinating insects and birds. And in some cases, it's not just about color. There is really lovely research from a researcher at the University of Bristol that show that on the surface of flower, you also have temperature pattern. And again, this is a cue that insects can follow. Heat is a reward for pollinator. They can't heat their body, so very often they will land in flower or even sleep in flower to keep warm. And these temperature patterns are not just providing heat. They're also helping guiding pollinator to the center. But pattern can also be used sometimes to deceive. And one of the most famous examples, I'm sure most of you have heard about it, this is the bee orchid. So these really do a really good job at mimicking the body of a female bee. It's not just the visual appearance, but their hair. There is molecular mimicking the pheromone. It's an incredible product of evolution. And the male bee tend to hatch before the female, and when they see that, they get really excited. They think it's the female. They go trying to mate with it and provide pollination services for the orchids. In this case, it's a bit harder to find out what this flower mimic. And it's because it's not a flower. It looks like an orchid, but it's actually an orchid mantis. But I always use this example because it shows that when something is good, other things try to copy it. So flower patterns are so good at attracting insects that this mantis, this prey mantis, mimic the pattern and the deposition of color of the orchid flower. It sits in the orchid plant, you think it's a flower, pollinators come and land on it, and then I never tell the rest of the story because it's not nice. Okay, more recently there had been a bunch of research that's really exciting because although patterns are super important for pollination, people have realized they can do more than that. And I'm going to show you two examples. So if you remember the silverweed I was talking to you about, researchers in the US collected silverweed of different populations, so either very high in altitude or very low, very close to the equator or very close to the pole. And what they found is that the size of the bullseye, the size of the pattern vary. And they found that, yes, local pollinators had developed preferences for the local proportion of the pattern. But it's not what explains the distribution. What explains the distribution better is the amount of UV the plant is receiving. So this pattern is a bit like sunscreen. If you grow really close to the equator or really high in altitude when you are subjected to lots of UV, you tend to have a big bullseye. How does that work? Well, if you think about it, in the center of the flower, you have the most precious part of the flower. You have the reproductive organ, like pollen grain. And if you have a small UV-absorbing bullseye and you grow in a place with high UV level, the UV will bounce on the periphery and reflect back on the pollen, and this really damages the viability of the pollen grain. So by having a bigger bullseye, you shield the pollen grain from that. You absorb the damaging UV ray. Same thing with sunflower. Again, this is the same species of sunflower. They are wild sunflower, not the one we grow as crop. And again, people found an impressive variation of bullseye size. In that case, they realized that it was an adaptation to desiccation. The more the plant grows in a dry environment, the bigger the bullseye. And this is because the molecules that absorb the UV, they're also very, very good at retaining water in the petal. So they help the flower not completely crumbling when it's hot. And this can be really useful for us also and to understand and track events. So the same team that work on silverweed managed to exploit samples from herbaria, because when you keep the flower, you can still visualize most of the pattern. And they imaged the size of the bullseye in herbaria sample collected a long time ago, and they went back in nature and tried to collect the same species in the same location. And long story short, what they found is that there is a very good correlation between level of UV going up, ozone going down, and the size of the bullseye increasing. So you can kind of use this pattern as marker and tracker of climate change and see how plants are responding to this variation. So if we recap, yes, patterns are very important to attract pollinators, but they also help them orientating themselves and performing pollination efficiently after landing, but they also fulfill all sorts of what we call abiotic function, so helping plants face and deal with environmental stressors. But it's not just about color and pigment. What we realized recently is if you image the surface of a petal with a scanning electron microscope, for instance, this is how these two images were taken, you find a huge diversity of cell shape on the surface and texture. What I mean by texture is that the surface of all aerial plant organ is covered by a waxy layer called the cuticle. The main function of this layer is just to prevent water loss, but it can actually be sculpted in all sorts of way and give the surface of leaves and petals interesting properties. So what we realized recently is that actually the distribution of this different cell shape and different texture is not random. It tends to coincide with part of the petal that also have different color. So what it means is that the plant must have a way of drawing boundary on the surface of its petal, divided into parts, and the cell in this different petal region will produce different pigments, but also different cell shape and different texture. Why is it? Why is there such a good correlation between the chemical part, the pigment, and what I call the structural part, which are cell shape and texture? Well, this is because actually these structural elements, they directly impact the appearance of the flower. I'm going to show you two examples. The first one, we've known that for a very long time, and it's, again, a snapdragon. So these two snapdragons, one looked paler than the other. When people extracted the pigment, they found there was no significant difference in the amount of pigment they produced, which was a surprise, as though they isolated the pigment mutant. But when they imaged the surface, they realized that instead of the conical cell, the cell had been completely flattened. And this matters because when you are conical, you act as a lens, and you focus the light in the vacuole that contain the pigment, meaning that you get the most color intensity for the amount of pigment you've got at the center of the cell. When the cell is flat, this doesn't happen, meaning the color appear much duller. So this shows how cell shape modify color intensity. And this is also true in the UV. Okay? These are examples of UV petal pattern. A team of physicists in Germany, what they did is they took petal with UV pattern, they pressed them in dental wax, made a copy with epoxy resin, so you have a plastic petal that doesn't have any pigment but replicate the surface. And then they image this plastic copy, and they could still see the bullseye, they could still see the pattern. Not as strong, but they still see, you can clearly see that there is a region that appears darker. This means that the UV pattern is not just created by molecules that absorb UV differently in the cell, but also created by the difference in texture. And indeed, in this plant, for instance, they noticed that you have conical cell in the two regions, but the aspect ratio, the size and the width of this conical shape vary, and this contributes to the difference in UV absorption. So these are examples of how structure modifies color, but structure can also create color from scratch. This is a species we've got growing in the botanic garden in Cambridge. It's a little daisy from South Africa. It's called Orsinia. And we tried for a long time to extract the blue pigment at the base of this petal, and we couldn't find blue pigment. And we couldn't because there is no blue pigment. And actually, what creates the color is not a pigment. We know that it's possible to do that. We've known that for a long time. These are two non-biological examples of perfectly transparent material that can create color, soap bubble and CD. I know nobody look at CD anymore, but this is what a CD is. How does it work? Well, you can make color if you are sculpted at the microscopic scale and you create structures that are very regular and so tiny they can split the light and separate the different wavelengths of light. So the tricks here is that you just separate the different wavelengths and you reveal the color of the rainbow. How does it work in the case of a CD? Again, I know you'll never do that, so I don't want you to miss out. If you take a CD and you look at it under an SEM, you'll see this combination of what we call ridges or striation and grooves. These are the data grooves. They are regularly spaced. They are about the same order of magnitude as the wavelengths of light. So what they do, they act as what we call a diffraction grating that splits the different components of the white light. Now, this is important because this means that the color created with this structure have unique property that you can't obtain when you use pigment. The first one is they vary because the color you see will depend of the angle at which you look at the object. Okay, that's what we call iridescence. The other property is that very pure, very intense, so you can see them from a long distance. And also they can last forever. Okay, and this is probably why both plant and animal have used this structural trick to create color. More for butterflies, super blue, intense, very, very strong signal that can be seen from kilometer by another butterfly. Completely structural color, no pigment involved. No blue pigment, I should say, involved. And indeed, what we realize is in the case of our daisy, that's exactly what happened. The surface of the petal is covered by striation of the cuticle. You know, this waxy layer is not smooth, it's striated. And this striation, they split the light. Now there is a trick. The light is not split perfectly like a CD. There is defect. You can see the striation. They are not as neat and as nicely organized than the one on a CD. And the effect of this disorder is that it shifts the effect toward the blue UV part of the spectrum. And you would see, well, it's just because plants are not very good, they can't do perfection. No, it's not that. Because we keep finding examples of this across the angiosperm. It's always messy in different way, but the effect of the disorder is always to shift toward the blue UV. And if you remember what I told you earlier, UV, blue are things that pollinators see really well. So we think it's not an accident. We think actually a bit of disorder is good in that case. So I'm not going to tell you more about that for now, but I'll come back to it later. We think it's widespread iridescence, is created by a bit of disorder, and it provides cue that animal can detect and efficiently, and it can help them forage more effectively. It's not about just visual signal. We know this is SEM images of a feet of a bee, okay? And you can imagine that if you have to walk on this texture, the surface of the petal matter. And indeed, researchers have shown that having conical cell is a very good idea. If the weather is still and there is no wind, it doesn't really matter. But now, if the flower is being shaken, the pollinator will prefer plants that provide grape with conical cell. And this explains probably why it's not random. If you look, this is the flower of field beans, so it's like a legume flowers. It's got all these different petal type. When you look at where the conical cell, it's not random. It's exactly on the part of the flower where the pollinator establishes contact. So it's really providing gripping to the pollinator. So to sum up, microscopic patterns created by different in texture and cell shape are important. They can change the intensity of the color, but they can also create color from scratch. And they can also modulate the capacity of a petal to absorb or reflect UV. And they can provide grips, but they can also provide the surface of the flower with all sorts of other interesting properties like superhydrophobicity. I won't have time to go in today. So, okay, I hope you're convinced that petal patterns are really important. But the thing that's very sad is we still have no idea how flowers create this pattern. So plants have been flowering for 150 million years. They do that every day without us doing anything or paying attention, and we still don't know how they do it. So this really bothers me. So I want to know how they do it. And in order to do that, the first thing we needed to do is to find a suitable species. Why? I love Arabidopsis. I've done my thesis with Arabidopsis. It's a fantastic model system. But if you're interested in petal pattern and interaction with pollinator, that might not be your best choice. Flowers are really tiny. They self-pollinate. They don't really rely on pollinator. So we developed an alternative to Arabidopsis. And it's a species of hibiscus. But I really like to call it the Arabidopsis of hibiscus because it's a weed. It grows really fast. So it's not like the big red hibiscus you can buy in a garden center. And it ticks really all the boxes for me of what makes it a good model system. It's very hard to kill even if you forget to water it. It makes lots of flower, lots of seed. It's self, but you can cross it. It's pollinated by animal. It's very close to important crop species like cotton and cocoa. Very disappointing nobody picked up chocolate. As I would definitely save chocolate as my seeds in the seed bank, I've decided. And importantly, we can manipulate it genetically. So we can mess up with gene and see what happens. Another reason that makes it a good system for us is that it's got a simple petal pattern. To start understanding how plants make pattern, let's start with simple. So by simple, I mean it's a bullseye. So you just need two regions, okay? The base of the petal different from the top. And I'm sure you can see they are different because they have different colors. It's purple at the base, is white at the top. If you image them with the microscope, so the big images are SEM images, the little images in the corner are the same petal but imaged with a light microscope from underneath. So what you see is the white cell, they are smooth and conical, okay? So they grow and they form the little cone. In the purple part, it's a completely different story. The cell remain flat, they grow, but they grow along the petal surface, so they become really long, elongating rectangle. You can't see them with the SEM. We can't see the outline of the cell really well because they are covered by this striation of the cuticle, okay? And these striations are continuous between cells. But if you image it from underneath with the light coming underneath, you don't see the striation, but you can really see the shape of the cell. And this striation, they create this kind of blue metallic sheen you see on top of the purple pigment. So without the striation, it will look like that. With the striation, it creates this blue halo on top of the petal. So we thought this is great because this is a simple model where there is two very different type of cells, different color, different shape, different texture, divided by one boundary. So it's a nice system for us to start understanding how plants create patterns. And the last reason that makes it a very good model system is it comes from New Zealand and Australia, and it's part of a small group which look fairly similar when they are vegetative, so before flowering. But when they flower, they show a huge range of variation in the bullseye. So we thought it's a very nice system if we want to understand how evolution manipulates developmental process to create diversity of pattern. And indeed, we recently, so two people in the group, Joe was the first postdoc of the group, and Holly was an undergrad that ended up coming back for a thesis, so she's now a doctoral student in our team. They solved the relationship between this group because it's an example of recent speciation, so we didn't know the relationship. And what they found is that actually the bullseye keeps shrinking, keep getting smaller during the evolution of the group. And I'll come back to that later on because we've now understood why. So what we try to do in our team is we try to understand what are the genes that are important to create pattern, how do they work together as part of gene regulatory network, and how the activity of this gene is going to change the behavior of the cell, and how this behavior is coordinated at the tissue scale. So we try to go from the tiny, tiny molecule to the cell and to the tissue level. And then, of course, we want to understand how evolution tinker with this process to create diversity. And finally, we try to see what pollinators think about these changes. If you mess up with the pattern, how does this impact pollinator behavior? So I'm mostly going to focus on the developmental aspect, but at the end, I'll have time to go a bit over evolution and behavior of pollinator to give you a taster of what we do. So how do you start understanding how development of petal patterns? So we didn't really know where to start, so we started with simple. What we did is we just dissected flower bud and looked at the petal surface. And what we found, we created some artificial division of development into stages. And we found that things start very early on. So the petal is about 4 or 5 centimeters in the mature flower. What we call stage 1 is a stage when it's very tiny, it's 2 millimeters tall, and it's completely green. So this is our starting point because it looks on pattern. Okay, nothing had happened at that stage. The first thing that appears is the pigmentation very early on. You can see that the petal is divided. There are pigment, non-pigment. But if you look at what the cells are doing at this stage, they are not doing very much. They are still actively dividing. They don't have a specific shape. They don't have a specific texture. This difference in shape and texture, they come much later. Okay? So this was interesting to us because it means either everything is set into motion really early on, but for some reason, it's easier to make the pigment, it takes longer to create the structural things, or these two programs are activated at different time points, but they need to be coordinated because you need to switch cell shape and texture at the same position that you switch pigment production. So we did lots of things to try to find the genes that were important. We did lots of things where we didn't have to make assumptions, like we did a mutant screen, we'd compare gene expression. And we did things where we thought, okay, we don't really know what's happening in hibiscus, but people have studied plant development for a long time. We know, for instance, that auxin is important, so let's see if auxin has something to do with petal formation. I'm going to talk about almost none of these things. I'm just going to mention one thing we did. So one thing we did was to use comparative gene expression to identify a gene that might be important. The idea was, okay, some of the genes that are important to make the pattern, they must be expressed differently between the base and the top of the petal. Okay, otherwise the cell would not end up looking different. So what we did, and this was done by May, a PhD student in the team, she dissected hundreds of tiny petals, chopped them in half. We couldn't see anything, so we just cut them in the middle, extracted RNA, and we did bulk RNA-seq to identify the population of RNA in the white part, or what will become the white part, and RNA, so gene expressed in the purple part. And we did that 5 different times independently. So we obtained 10 different transcriptomes, 5 for the top, 5 for the bottom. The first thing you can do is you can compare these transcriptomes with each other. And what we found, so the number represents different transcriptomes, and we are comparing them with each other. You do all the pairwise comparison. What we found, and we were really surprised, is that actually the 5 transcriptomes that come from the bottom, they look very, very similar and very different from the 5 from the top. So this means that although it looks like this petal is not doing much, and the cells look the same across the surface, they are not the same. They already know where they are, and are already expressing very different sets of genes. Another way of representing the data is these volcano plots here. So you can see each dot is a gene. Purple genes are the one more expressed at the base. White dots are more expressed at the top. And we found about 1,000 genes more expressed in the purple, about a third of that more expressed in the white. So this means that, okay, cells in the white and the purple region, in the future white and purple region, are already expressing different genes. But how do you know you got the right gene? Because it's a lot of genes. We're talking about 1,500 genes. So what we decided to do 2 things, all based on gene manipulation of gene expression. First, you can force a gene to be expressed in the wrong place. So you take a gene that normally only expressed at the base of the petal and you force it to be expressed at the top. Or you can break a gene and see what happens when the gene is not working. So this is exactly what we did. How you do that in hibiscus? Well, we germinate the seeds overnight, we chop the little stem into pieces, and then we infect them with a bacteria that contain a genetic construct that allow us to either express a gene everywhere or knock it out with CRISPR. Then it's a system of tissue culture, meaning you put this little piece of stem on media with different hormones. The cell, they differentiate, they go back to stem cell, and then by tweaking the hormone you use, you can recreate a full plant from a transformed cell. and then you get a new little plant and you can move it in the greenhouse. How do we know they've been transformed? We always squeeze in a fluorescent gene, a gene that codes for YFP, that go to the plasma membrane. So this means that we can identify transformed plant really easily just by looking under the fluorescent microscope. And it also goes to the plasma membrane, which allows us to track cell, and that's really useful. So this is the full process, and it takes a few weeks, of course, it's not immediate. But it allows us to really manipulate gene expression and understand what the genes are doing. And what we found is that many of the genes we identify like that were actually important to make the pattern. We found genes that mess up the color. We found genes that mess up the texture. Like here, we lost the striation, for instance. And we found genes that change the shape of the cell. So here, the conical cell become flat elongated, like the cell at the base, or they become puzzle cell shape, like what you have on leaves sometimes. So what was our take-home message from that? Well, we identify all sorts of genes. We are recreating the gene regulatory network. But the important thing is that plants use different sets of genes to control these different features. There are genes that do pigment, there are genes that do cell shape, and genes that do texture. But we also got a different kind of plants. We got manipulated line in which everything was fine. So the characteristics of the cell were okay, but the proportion of the pattern were different. So they were either smaller or they were bigger. And this means that there is a way for the plant to control everything and to slide everything at once. Okay? So all the characteristics can be shifted. And this, combined with other experiments I'm not going to tell you today, made us realize that actually, when I told you our starting point was stage 1, it was completely wrong. Okay? Stage 1, the petal has already been divided. So everything that's interesting happened in the shadow, happened very early on when the petals are absolutely minute, and they look super boring, super green. But actually, that's where all the important biology happens, because this is where the cell learn where they are on the petal, and this is where they make different decisions. So, and we know that this can only happen because there is a signal that comes from elsewhere in the plant. And what we're trying to do now is to understand what is this signal, and we are interacting with all sorts of small molecules that are interesting. So hopefully next time I come, I'll have some update on that. So all that is fine, but I told you we don't want to just identify gene. Identify gene is nice, you can do arrow between them, but it doesn't really tell you you've understood much about what's happening. So what we're trying to do is we work a lot with a team that do computational simulation to understand how this network of genes evolve, but also how do they affect cell behavior. And what we're trying to do at the moment is combine both this simulation and experiment to see what is happening during this pre-patterning phase to create a boundary, and what is happening afterwards. Imagine you set the pattern very, very early on with the petal is very tiny, and then the petal is growing to grow enormously, but the proportion remains the same. And this is a real challenge for the plant to maintain that. This is ongoing, so although instead of telling you about things where we have no answer, I'll briefly show you an example where we use modeling with experiment and we got some useful answer. And this is actually why I started working on hibiscus. What I really wanted to do is understand how the cell creates the striation. I can understand how the cell controls what's happening inside, but how do you control what's happening on your head outside? And so we talked to physicists, and they told us, oh, this is really easy. It's called mechanical buckling. It's what happened. I'm sure you've all done that. If you take a piece of cling film to put food on, you stretch it, it forms striation, right? It's because with mechanical stress, you create instability, and the instability, the material deal with the stress by wrinkling. So we thought this is great because you can control the type of pattern you get with inorganic material by deciding how much you stretch and which direction you stretch. So we thought if this is what's happening in plant, this gives us a good idea because the striation form when the cells start stretching. So it means that in order to control whether it forms striation or not and what type of striation the plant forms, the cell needs to control two things. It needs to control how much the cell grows and how much of the material is making, how much of this cuticle is being produced. And we produce a really nice model that allows us to predict if you know how big a cell is and how much cuticle it makes, I can tell you what type of texture. And this worked really well, except we keep finding cases where it didn't work. And I'm just going to show you one case. So this is wild type, you remember, you have this striation that make the petal appear bluish. We produce a transgenic line. I'm not telling you exactly what we did, but with modified transcription factor, this line is completely smooth. There is no more striation. But the cells have exactly the same size. And when we look at the cuticle, these are cross-section when we stain the cuticle in red. the cuticle is exactly the same thickness. If our model was right, this is not possible. Because if you have the same cell size, the same cuticle thickness, you should have the same texture. And what we realize is that we completely overlook one important thing. We completely overlook the chemistry of the cuticle. So the cuticle is a polymer of fatty acid called cutin. It's created like a mesh. And in the gap of the mesh, you can put all sorts of small molecules. And these molecules matter. They act as nanofiller. They can be wax, terpenoid, whatever you want. And they are going to change the mechanical property of the cuticle, meaning that some cuticle might be more able to wrinkle than others, just based on what they are made of. So we team with chemists, and they develop a protocol that allows us to measure, analyze the composition of the cuticle in different spots of the petal. We did exactly that. And what we found, this graph is just separating the compound we identify based on their chemical properties. So every symbol is a chemical. All you need to know is that we found some compound in both regions. They are the little pink triangle. But we found many, many compounds only in one region and not the other. This means that although the cuticle is continuous, it is just one organ, it's completely different chemistry in the two parts of the petal. And indeed, we did this experiment again and again, both on transgenic light and on natural variants, so species that naturally have striation or don't have striation. And what we found is if you use a principal component analysis to separate the data, you can really resolve the species. So actually, if you want to predict if the striation will form or not, it's much better if you look at the chemistry of the cuticle. It's a better predictor of what the plant can or can't do. So the idea was like, okay, the chemistry is different, the mechanical properties should be different, but we didn't really test, is that true? Are we just making this up? And this is where we work with Sarah at the Sainsbury Lab. And Sarah developed this really cool tool that I'm sure you've heard about or you will hear about. It's a confocal microextensometer. So it's basically little, Sarah, close your ears because I'm going to say all sorts of horrible things. So it's basically like a mini system of like two little arms. You can stick your sample to it, and then you can decide how much the arms are going to pull away. So you can stretch your sample. You know exactly how much force you apply to stretch it, and you can record that, and you can also image the surface, so you see exactly what happened. So what we did is we pick up a petal, a piece of petal just before the striation form. If you take this petal in a wild-type plant, so before it has no striation, you stretch it. Stretching it is enough to create the striation. I remember when I tag along when Sarah did that, it was magical. You see the striation appearing just almost instantly in response to the stretch. But if you do that with the other line, the line that remains smooth, you can stretch it as much as you want, it never striate. So this really shows us that this difference in chemistry change the behavior of the surface. And you can really see that they respond, they are just different material. So take-home message is that we start to finally understand a little bit about how plants create this pattern. And what I want to tell you in the last few minutes is how we can use this knowledge to also understand how evolution is changing the pattern and help us understand better how pollinators interact with this pattern. So I told you the first thing we knew is that we were really puzzled because it looked like the bullseye kept getting smaller and smaller, and we didn't know how. So we decided to, okay, let's take a look at the sister species of Hibiscus trianum. It's called Hibiscus richardsonii. It's got a bullseye, but it's very, very small. So we thought, okay, sister species is quite close to our species. And we thought, let's see if we can figure out what's happening there. So the first thing we needed to do is to go back to development and identify what genes are important to make the pigment and make sure that you only make pigment at the base and not at the top. We found some gene, and we were able to show, and this was work done by a PhD student, Alice, that used to be a Gatsby student as well. So these are all transcription factor. Normally, the cream transcription factor is expressed mostly at the top. If you force it to be expressed everywhere, you get paler and paler bullseye. So this suggests that cream is a repressor of pigmentation. We found two genes, we named them berry, also transcription factor, mostly expressed at the base. If you force them to be expressed everywhere, you get pink flower. So these are activators of pigmentation. And combining this kind of experiment with others, we were able to create a little network, gene regulatory network, that explains why you only make the pigment at the base. Why is that important? It's important because it gives us a starting point to try to understand what evolution may have messed up. And what we found is that the berry gene, the berry1 gene, has been broken up in richardsonii. So there is premature stop codon, a big insertion. The gene is still there, but it's broken. And this is why you create a smaller bullseye. things became really interesting when we started looking at the other case of smaller bullseye, because evolution is very not creative. The same gene has been targeted in this other species, but there the gene is completely missing from the genome. So it's a completely different process, but the same gene that's been targeted to reduce the bullseye. Does this matter? Do bees care about the change in bullseye size? Well, we decided to ask them. So how do you test what a bee is thinking? It's actually quite easy. What we do is we use colony of bumblebee, and there are special colonies, so they are much smaller than real colony. They are research colony, they have a queen and about 40, 50 workers. It's just to make our job easier. And we connect them to a flight arena, which is a giant box with transparent top, so you can see what's happening. And then there is a tube, a gated tube, so you can decide who comes in, who comes out. We label every single bee, so we can track every single individual. So the first question was, if I give you two flowers, one with a small bullseye, one with a big bullseye, can you see the difference? Because if they can't see the difference, then there is no point doing any further. They will look the same for them. How do you know if they can tell the difference? Well, you can do that in different ways. This is the way we use. So you can take a bunch of small bullseye, small of big bullseye flower. You associate one type with a reward, sugary water, bee like it. And then the other type, you associate it with what I call a disappointment. It's just flat water. It's not that they don't like it, it's just not very nice. There is nothing interesting for them in it. What is the idea? The idea is at the start of your experiment. So imagine you have 10 flowers, 5 of each. You let a bee go in. This bee has never seen flower in her whole life. Doesn't know what is what. So the bee should land randomly, and it should be 50-50. If the bee can tell the difference, it should really quickly learn that only one type, big or small bullseye, has the sugar. So what you expect to see is your bee will just stop visiting one type and will only visit the good type. If the bee can't tell the difference, it's got no choice but landing and testing to figure out if it's sugar or not. So the bee should keep choosing randomly. So it looks something like that in terms of data. On the x-axis, you have the number of choice the bee is making. On the y-axis, you've got how good it is at choosing right. And at the beginning, you start 50-50. If you can see the difference, the bee learn, get very good at it. If there is no ability to see the difference, you remain at 50-50. So when we did this experiment, we said, okay, we are not going to do it with real flower to begin with, because these are two different species. They're very close, but two different species. They might differ in scent, their shape might be a little bit different, there might be all sorts of things we have not think of. So we did something very basic. We 3D printed discs that mimic the proportion. It's not fancy, but it's very. You can do lots of things with simple things. So we had 5 with a bullseye that mimic the size of Trinum, 5 of Richardsonii. And we did this test. Sometimes we put the sugar on one type, sometimes we did on the other type. It's a super easy task for bee. They were almost laughing at us, thinking, yes, of course I can tell the difference. Good. So you can tell the difference, but do you have a preference? Or you just can see them differently? So we did different experiments for that. I'm only explaining the simplest one. The simplest one is you take 2 discs, a small bullseye, a big bullseye, both with sugary water. You put them at the back of the flight arena, exactly at the same distance from the entrance. You let one bee, and you just record what the bee is doing. This bee has never done an experiment, never seen flowers, and you do that with many, many, many bees. So it looks something like that. You let the bee fly out, and it will go, fly, have a look, and decide which one it likes. And when we did that, we found that almost 3 out of 4 times they go for the big bullseye. We then repeated it with the real flower, and I was thinking, surely something will compensate it. No, the preference is even stronger. So it's 8 out of 10 times they go for the big bullseye. So they really do not like the small bullseye, which brings all sorts of interesting questions for why do the bullseye keep getting smaller. So what we are doing now? Well, now we are really taking advantage of the fact we have a model system in which we can do the genetics. Because lots of what we know about pollinator perception is based on observation in the field that we hope to do, and that's very important. But you're really limited in what you can control or can't control. We've generated now a bunch of transgenic line or genome-edited line where we change one gene, and you know exactly what features of the bullseye you change. So we are now testing how different in size 2 bullseye need to be for the bee to perceive them different. If you are 5% bigger, is that enough to trigger different kind of response? The other question we are investigating, I'm really interested in, is this interplay between pigment and structure. This is one of my favorite transgenic lines. So this was an accident. We were trying to express everywhere the enzyme that makes the pigment. But because the gene that codes for this enzyme is already expressed at high level, When we force it to be expressed even more, we had exactly the opposite. We silence everything, which was annoying at the time, but actually very useful because there is no more pigment in this plant. But you still see the bullseye because the cells at the base are different from the cell at the top. So the way the light interferes with the material is different. And the question is, can bees see the bullseye without pigment? The answer is yes, they can see it very well. So we are trying to create flower when there is a just purely pigmented bullseye or purely structural and see which one matters most. Okay, so conclusion. I hope I convince you that really flowering plants are incredible engineers. They can create everything. You know, I always start my talk normally by saying plants don't run away from danger. And it's not because they can't, it's just because they don't have to. They are too smart for that. They can just manipulate their surface to deal with whatever happens without having to running away. And this microscopic feature, so whether cell shape, texture, but also pigment, really give them interesting properties. And we are only starting to understand what these properties are, mostly because it relies on biologists working with physicists and chemists. So interdisciplinary work is really important for this type of work. And finally, by combining all this approach, whether it's genetics, computational modeling, mechanics, we can start understanding how these patterns are formed, how they evolve, what pollinators think about it. And importantly, the question we're trying to answer, they are not just plant-specific. It's about how cells make decisions. And you are only here because at some point your first cell took decision to divide, and your cell became to differentiate. So it's also a question that applies to any form of living organism. So I love plants, and that's why I study plants, but they're also excellent model systems to ask any question about biology. And there is actually lots we understand. Basics of our genetics come from studying plants. So take-home message is consider plants even if you also like animals. It's not incompatible. Okay, thank you very much. These are the people that have done all the work. So these are the current people in the team. I mostly talk about May's work. Who did I talk about as well? I mentioned Holly and Joe that do the phylogeny. Briefly mentioned Yushi at the beginning. These are all the collaborators, so collaborators at the Sainsbury Lab, elsewhere in Cambridge, and also outside of the UK. Thank you for your attention.